Welcome to the Deep Dive, the show that's your shortcut to being well-informed. We take a whole stack of sources and really pull out the key nuggets of knowledge for you.
That's the plan.
Today, we're diving straight into something truly incredible,
Mm-hmm.
your own human brain. I mean, just think about it for a second. How do you experience the world around you? How do you stay awake or learn something new or feel emotions or even talk?
It's staggering, really.
It's this, amazingly complex symphony of processes all happening inside your head right now.
Mm-hmm.
So our mission for this deep dive is to pull back that curtain on what the textbook calls higher order functions. You know, the really intricate ways your central nervous system processes information.
And we're focusing specifically on Chapter 8 of Vander's Human Physiology. We'll distill it down, give you the essentials without hopefully overloading you, just what's most important to grasp.
Right, building on what you might already know about, say, basic nerve signals,
Mm-hmm.
chemical gradients, action potentials, that sort of thing.
Exactly. We want to connect those basic building blocks to the big stuff. Conscious attention, motivation, learning, memory, language, it all starts there.
And a core idea here, which the chapter really emphasizes, is how this information flow between cells, tissues, organs. It's fundamental for homeostasis, right? For integrating everything.
Absolutely fundamental. It ties everything together.
Okay, so let's get into it. First big topic, states of consciousness,
Yeah. And the book makes a useful distinction right off the bat.
which is,
Well, first, there are the states of consciousness. Think of it as your level of alertness. Are you wide awake, kind of drowsy, or fully asleep?
okay, the overall state.
Then there are the conscious experiences. These are the specific things you're aware of within a state. So your thoughts, feelings, what you're seeing, maybe a dream if you're asleep.
Got it. State versus the content of that state. And how do we measure these states?
The key tool here is the electroencephalogram, the EEG.
Ah, yes, the brainwave test.
Pretty much. It's basically a recording of the electrical buzz on your scalp picked up by electrodes. It's how we define these different states of consciousness.
And what's it actually picking up? Not individual neurons firing.
No, not single action potentials. It's mainly reflecting the combined synchronized graded potentials. Think summed postsynaptic potentials from hundreds of thousands of cells in the cerebral cortex. Pyramidal cells, mostly. It's the big picture activity.
Okay, the collective hum. And these EEG waves, they have properties.
Two main ones, amplitude and frequency. Amplitude tells you how much similar electrical activity is happening at the same time. Big amplitude means lots of neurons are firing in sync.
More synchrony, bigger waves.
Exactly. And frequency, that's how fast the wave is cycling, measured in hertz. Generally, lower frequencies mean less responsive states, deeper sleep, for example. Higher frequencies usually mean more alertness.
And where do these waves come from?
Well, current thinking points towards clusters of neurons down in the thalamus being really important. They seem to generate these rhythmic oscillations that then influence the cortex.
Interesting. So the thalamus is like a pacemaker for these rhythms.
In a way, yeah. Clinically, the EEG is super useful. Monitoring patients under anesthesia, diagnosing neurological issues like epilepsy, determining coma, brain death.
You mentioned epilepsy. How does that show up on an EEG?
Epilepsy is characterized by these really abnormal, highly synchronized bursts of activity in the brain. On the EEG, you see these distinctively large amplitude waves, sometimes sharp spikes. And this corresponds to the behavioral changes, muscle contractions, or loss of consciousness someone might experience.
Okay. Now let's talk about the waking state. Our brain waves aren't just one type when we're awake, are they?
No, definitely not. When you're awake but relaxed, maybe with your eyes closed, you often see the alpha rhythm. That's about 8 to 12 hertz, usually strongest over the back of your head. It's linked to decreased attention, kind of a relaxed, idling state.
But then if I open my eyes or start thinking hard,
Then that alpha rhythm tends to disappear, replaced by faster, smaller beta rhythms, anything above 12 hertz. This shift is called EG arousal.
EG arousal. And that's tied to paying attention.
Exactly. It's linked more to the act of attending rather than just perceiving something. And then there's even faster activity, gamma rhythms, like 30 to 100 hertz.
That sounds fast.
Yeah, and these often spread across wider areas of the cortex. They're thought to be involved in binding different aspects of an experience together, like linking the sight and sound of something into one coherent perception.
Cool. Okay, so that's being awake. What about sleep? It's not just off, is it?
Oh, far from it. Sleep is an active, cyclical process. As you get drowsy and fall asleep, your EEG changes dramatically. You go from beta through alpha, then into slower theta waves, 4 to 8 hertz, and finally the really slow, large delta waves, less than 4 hertz, in deep sleep.
And behaviorally, you relax, it's harder to wake you up.
Right. Your muscles relax, your threshold for sensory input goes way up, less motor output.
And there are different phases, NREM and REM.
Correct. Two major phases. First is NREM, non-rapid eye movement sleep. This itself has stages N1, N2, and N3. You basically descend through these stages.
Getting progressively deeper.
Yeah, the EEG gets slower and the amplitude gets bigger as you go from N1, where theta starts mixing with alpha, through N2 with its sleep spindles and K complexes, down to N3, which is dominated by those big delta waves. That's often called slow wave sleep.
Okay, deep NREM sleep, then comes REM.
Usually, yes. After cycling through NREM, maybe back up to N2, you typically enter REM sleep, rapid eye movement sleep. And this is why it's called paradoxical sleep.
Paradoxical, why?
Because even though you're deeply asleep, your EEG looks remarkably like you're awake and alert. High frequency, small amplitude waves. Your brain's oxygen consumption is actually higher during REM than when you're awake or in NREM.
Whoa. And that's when we dream most vividly.
That's typically when the most memorable story-like dreams happen. Another key feature of REM is muscle atonia. Your skeletal muscles are actively inhibited, very relaxed, except for your eye muscles, hence the name, and your respiratory muscles, thankfully.
So you don't act out your dreams.
Pretty much. Keeps you safe in bed.
How often do these cycles happen?
If you sleep undisturbed, it's roughly a 90 to 100-minute cycle, repeating maybe 4 or 5 times a night. You tend to get more deep NREM sleep earlier in the night and longer REM periods towards the morning.
And why do we even need sleep? It takes up so much time.
We still don't know everything, but it's clearly a fundamental biological need, a homeostatic requirement. It seems crucial for your immune system, for cognitive function, learning, and especially memory consolidation.
Memory consolidation, like filing away the day's events.
Sort of. It might involve clearing out metabolic waste products that build up while you're awake, reactivating important neural pathways, and strengthening the synapses involved in learning.
So what's controlling all this? The switching between wake and sleep.
It's a complex network. A big part is your internal circadian rhythm, that daily cycle. The master clock for this is the suprachiasmatic nucleus, or SCN, in the hypothalamus.
LCN.
The SCN helps time things. For example, it activates certain neurons in the morning that release orexins, which promote wakefulness. At night, it signals for melatonin release, which helps lower body temperature and prepare for sleep.
Orexins.
Yeah.
Aren't those linked to narcolepsy?
Exactly. Orexins, also called hypocretins, are neuropeptides crucial for maintaining the awake state. They stimulate other wake-promoting areas. A lack of orexins leads to narcolepsy, characterized by that unstable switching into sleep.
And what about the general wake-up signal?
That involves the reticular activating system, the RAS. It's a network of nuclei in the brainstem and hypothalamus that project widely up to the cortex and thalamus.
The RAS keeps us alert.
Essentially, yes. Ascendant RAS pathways release neurotransmitters like norepinephrine, serotonin, histamine. These act as neuromodulators, basically turning up the volume on excitatory activity throughout the cortex, making you more alert and responsive.
So orexins help keep the RAS going.
Yes, orexin neurons strongly stimulate those RAS monoaminergic neurons.
Okay, so how do we fall asleep then? Is it just the wake systems shutting down?
Not entirely. It's an active process, too. There's a sleep center, specifically, neurons in the ventrolateral preoptic nucleus, or VLPO, also in the hypothalamus.
VLPO. And what does it do?
These VLPO neurons release GABA, the main inhibitory neurotransmitter in the brain. They inhibit the orexin neurons and those monoamine RAS neurons, so they actively suppress the wake systems.
Ah, so that's why GABA-enhancing drugs like Valium make you sleepy.
Precisely. They tap into that natural sleep-promoting mechanism. It's a flip-flop switch. Wake systems active, sleep systems inhibited, and vice versa. Things like adenosine, a chemical that builds up the longer you're awake, also help tip the balance towards sleep by inhibiting orexin neurons.
Adenosine. Isn't that what caffeine blocks?
That's exactly right. Caffeine keeps you awake partly by blocking adenosine receptors, preventing that sleepiness signal from working as effectively.
Fascinating.
All right.
Okay, let's touch on the extreme ends. Coma and brain death.
Right. Coma is a severe, sustained loss of arousal due to significant brain impairment damage to cortex, thalamus, brainstem, or severe metabolic issues. No outward signs of mental function, eyes closed, no sleep-wake cycles, though intriguingly, some level of consciousness might persist in some cases.
And brain death. That's different.
Very different. Brain death is the irreversible cessation of all function of the entire brain, including the brainstem. It's the legal and medical definition of death.
What are the criteria? How is that determined?
There are strict criteria. Known cause of coma, ruling out reversible things like drug overdose or hypothermia. Then, tests showing absent in brainstem function, no response to pain, fixed pupils, no eye movement reflexes, no gag reflex, and critically, apnea.
Apnea?
Meaning no spontaneous breathing effort even when CO2 levels rise significantly after being taken off a ventilator for a period.
Huh?
It confirms the loss of brainstem respiratory control, plus other checks like repeating the exam after a certain time.
A very final, irreversible state. Okay, let's shift gears now to conscious experiences. What are we actually aware of?
Right. This is about the content of your awareness, sensory perceptions, feelings like fatigue or happiness, your thoughts, reasoning, even that sense of self.
And a huge part of this is selective attention, isn't it? Our ability to focus.
Absolutely crucial. It's your brain's spotlight, allowing you to filter out distractions and concentrate on what's important at that moment, like tuning into one conversation in a noisy room.
How does the brain decide what's important?
There's pre-attentive processing. Your nervous system is constantly evaluating incoming sensory info before it even reaches your conscious awareness, tagging things as potentially relevant and directing your attention accordingly.
And if something is repetitive and not relevant?
You get habituation. You stop responding to it. Think of a ticking clock, annoying at first, but soon you tune it out. That involves changes at the synaptic level, making the transmission less effective for that specific irrelevant stimulus.
What parts of the brain manage this selective attention?
It's a network. The thalamus is a key player, acting like a gatekeeper for most sensory information heading to the cortex. It can selectively filter what gets through, influenced by signals from both the cortex and brainstem.
The thalamus as a filter. Makes sense.
Also, the locus coeruleus in the brainstem releases norepinephrine, which acts as a neuromodulator, essentially boosting the signal strength of important stimuli relative to background noise.
And conditions like ADHD relate to this?
Yes, attention deficit hyperactivity disorder often involves difficulties with maintaining selective attention, impulsivity, and hyperactivity. Brain imaging suggests dysfunction in areas rich in catecholamines, like dopamine and norepinephrine pathways critical for attention and executive function. Medications like Ritalin work by boosting these neurotransmitters.
So how does the brain actually generate conscious experience itself? Where does awareness happen?
That's the million-dollar question, isn't it? The prevailing view isn't a single consciousness spot in the brain.
No magic consciousness center.
Probably not. Instead, it's thought to involve a temporary coalition of neurons, distributed across different brain regions, visual areas, auditory areas, memory circuits, emotional centers becoming functionally linked and synchronized for a period.
A temporary network, always shifting.
Exactly.
Yeah.
As your focus of attention shifts, the set of neurons involved in the conscious experience changes. The cerebral cortex is definitely critical. Damage to specific cortical areas reliably knocks out specific types of awareness.
Like that sensory neglect example.
Right. Damage to parietal association cortex can make someone completely unaware of one side of their body or the world, as if it just doesn't exist for them. It really shows how tightly consciousness and attention are linked. To attend is, in essence, to bring into consciousness.
Okay, let's move on to motivation and emotion. What drives our behavior?
That's motivation. The process is responsible for the goal-directed nature of our actions, often called drives.
And there are different types.
We can talk about primary motivated behavior, which is directly tied to maintaining homeostasis, like drinking when thirsty to restore water balance.
Basic survival drives.
Right. Then there's secondary motivation, which is more complex. It's influenced by things like habits, learning, social factors, emotions, incentives, maybe choosing a soda because you like the taste, not because you're actually dehydrated. The lines can blur, though.
And motivation is tied to reward and punishment.
Inextricably linked. Rewards are things we work for, punishments are things we avoid. These shape our motivations powerfully.
Where in the brain does reward happen?
Key pathways are the mesolimbic and mesocortical dopamine pathways. These originate in the midbrain and send dopamine to areas involved in emotion and decision-making, like the nucleus accumbens and prefrontal cortex.
Dopamine again. The reward chemical.
It's definitely a major player in reward and motivation. Those classic brain self-stimulation studies really highlighted this. Rats would press a lever nonstop to get electrical stimulation in certain brain areas, especially parts of the hypothalamus, if it triggered dopamine release. They'd even ignore food or water.
Wow, so dopamine signals this is good, do it again.
Essentially, yes, it reinforces behaviors. Drugs that boost dopamine, like amphetamines, are highly rewarding and addictive for this reason. Drugs that block dopamine receptors tend to be unpleasant or blunt motivation.
Okay, what about emotion? How is that defined?
Emotion is seen as this complex interplay between you and your environment. It involves evaluating situations pleasant, hostile, your internal feeling state happy, scared, and the resulting physiological and behavioral responses.
So 3 parts: evaluation, feeling, and response.
You could put it that way. Anatomical sites determine the emotional value, then there's the outward behavior, heart races, posture changes, and then the inner conscious feeling.
What brain structures are key for emotion?
The limbic system is central. The hypothalamus is involved in expressing emotional behaviors like rage responses, but the amygdala is really crucial, especially for fear.
The amygdala and fear. You mentioned patient SM.
Right, the patient with damage to both amygdala who couldn't recognize or feel fear. A striking demonstration. The amygdala interacts heavily with the hypothalamus, memory areas, attention networks. It helps assess threats and guide responses. The prefrontal cortex is also vital for experiencing and regulating emotions.
So the cortex helps us understand and manage our feelings.
Yes, it allows us to interpret emotions, connect them to causes, and modulate our behavioral responses. It gives us that layer of conscious control and understanding.
Let's shift now to altered states of consciousness.
Yeah.
What falls under this category?
These are states beyond typical wakefulness or sleepiness. Think hypnosis, experiences induced by mind-altering drugs, or states associated with certain psychiatric conditions.
Like schizophrenia.
Exactly. Schizophrenia is thought to involve profound disturbances in how the brain processes information. Symptoms are diverse hallucinations like hearing voices, delusions, disorganized thinking, emotional withdrawal, sometimes abnormal motor behavior.
What causes it?
The exact causes are still unclear, but it seems to be a neurodevelopmental disorder, possibly involving problems with how neurons migrate and connect during brain development. There's a genetic predisposition, but environmental factors likely play a role too.
And the dopamine connection.
Yes, the dopamine hypothesis is a major theory, suggesting overactivity in certain dopamine pathways, particularly the mesocortical pathway projecting to the prefrontal cortex. This fits with the fact that drugs increasing dopamine can worsen symptoms, and most effective antipsychotic drugs block dopamine receptors.
Okay. What about mood disorders like depression?
Mood disorders involve a loss of control over one's pervasive inner emotional state. Depression is characterized by persistent sadness, loss of interest or pleasure, low energy, anxiety, changes in sleep and appetite, sometimes suicidal thoughts. Brain imaging often shows decreased activity in parts of the limbic system and prefrontal cortex.
Are neurotransmitters involved here too?
Yes, major focus has been on the biogenic amines norepinephrine, dopamine, and serotonin. Most antidepressant medications work by influencing these systems.
Like SSRIs.
Right. Selective serotonin reuptake inhibitors like Prozac are widely used. They increase serotonin levels in the synapse. Older drugs, like tricyclics and MAO inhibitors, affect serotonin and undor norepinephrine through different mechanisms. The common theme is boosting these neurotransmitters.
But they take weeks to work, don't they?
That's a key point. The chemical effects are fast, but the mood improvement takes time. This suggests a more complex downstream effect, possibly involving changes in receptor sensitivity or even stimulating neurogenesis, the birth of new neurons, particularly in the hippocampus. Chronic stress, a trigger for depression, is known to inhibit neurogenesis.
So antidepressants might help rebuild brain circuits over time.
That's one leading hypothesis. Other treatments exist too, like psychotherapy and, for severe cases, electroconvulsive therapy, ECT, or repetitive transcranial magnetic stimulation, RTMS, which aim to reset or modulate brain activity patterns more directly.
What about bipolar disorder?
Bipolar involves swings between depression and mania. Mania is a state of abnormally elevated mood, high energy, racing thoughts, overconfidence, impulsivity, decreased need for sleep.
How is that treated?
The cornerstone treatment is lithium. It's remarkable because it helps stabilize both the manic and depressive episodes. Its exact mechanisms aren't fully known, but it seems to interfere with certain intracellular signaling pathways and might reduce excessive glutamate activity.
And this relates to psychoactive substances too.
Many psychoactive or recreational drugs tap into these same neurochemical systems, especially the dopamine reward pathways, to produce their effects, elevating mood or altering perception.
Like cocaine.
Yes, cocaine blocks the reuptake of dopamine, leading to a surge in the synapse, which is highly reinforcing. Repeated use can lead to tolerance, needing more drug for the same effect, and withdrawal symptoms when stopping.
And this can become a substance use disorder.
Right, which is the clinical term for addiction or dependence. It involves patterns of misuse that cause significant impairment or distress, often hijacking those natural reward and motivation circuits.
Okay, deep breath. Let's move into learning and memory. How do we acquire and store information?
Learning is fundamentally about acquiring information through experience, leading to a change in behavior. Rewards and punishments are really important guides in this process. Memory encoding is the set of neural processes that actually convert that experience into a storable format.
Are there different kinds of memory?
Broadly, yes. The textbook distinguishes between declarative memory and procedural memory.
Declarative is.
That's your explicit memory. Things you can consciously recall and declare or put into words. Facts, events, recognizing faces, knowing where you were yesterday. The hippocampus and other limbic structures are critical for forming these memories.
And procedural.
That's implicit memory how-to knowledge. Skills, habits, learned emotional responses. Riding a bike, playing piano, feeling scared of spiders. You don't necessarily consciously recall the learning process. This relies more on areas like the sensorimotor cortex, basal nuclei, and cerebellum.
So different brain systems for knowing that versus knowing how.
Exactly. And we also talk about memory duration.
Short-term versus long-term.
Right. Short-term memory or working memory holds information actively in mind for seconds to minutes, like remembering a phone number just long enough to dial it. It's crucial for ongoing thought and problem solving.
And long-term memory is the stored stuff.
Yes, information stored for days, weeks, years. The process of converting short-term memories into long-term ones is consolidation.
How does this work neurally? Is short-term memory fragile?
It seems to depend on ongoing electrical activity graded potentials or action potentials. That's why it's easily disrupted by things like head trauma, anesthesia, or shock, which can cause retrograde amnesia, losing memories from right before the event.
But long-term memory survives that.
Generally, yes. Once consolidated, it's much more stable. However, damage to the consolidation machinery itself, particularly the hippocampus and related limbic structures, causes anterograde amnesia.
Anterograde. That's the inability to form new long-term memories.
Correct. Patients can't transfer new experiences from short-term into long-term storage. They remember their past but live in a perpetual present, unable to retain new declarative information for more than a few minutes.
Like patient HM.
He's the classic case. After removal of his hippocampi to treat epilepsy, he has severe anterograde amnesia for declarative memory. He could still learn new procedural skills, but wouldn't remember having learned them. It powerfully showed the hippocampus's role in consolidation, not long-term storage itself.
What's happening at the synapse level during memory formation?
It involves plasticity, the ability of synapses to change their strength. A key mechanism is long-term potentiation, LTP, where synapses become more effective, transmitting signals more easily after intense stimulation. There's also long-term depression, LTD, weakening synapses.
So synapses get stronger or weaker.
Yes, and long-term memory involves more lasting changes, like new protein synthesis, alterations in gene expression, even structural changes, growing new synaptic connections, or changing the shape of existing ones. The brain physically rewires itself to store information.
Incredible. Okay, final section. Cerebral dominance and language. Our brain hemispheres aren't identical, are they?
Not functionally, no. While they look similar, there's significant hemispheric specialization. Most obviously, the left hemisphere controls the right side of the body, and vice versa for sensory input and motor output.
But language is really lateralized.
Highly lateralized for about 90% of people. The left hemisphere is typically dominant for the technical aspects of language, understanding grammar, producing words, speaking, writing, even sign language.
And the right hemisphere, does it do anything for language?
Yes, it's generally dominant for the affective or emotional aspects of language, understanding tone of voice, interpreting emotional cues, appreciating sarcasm or humor.
So left is the what, right is the how it feels.
That's a decent simplification. Key language centers are clustered in the left hemisphere, near the Sylvian fissure.
Like Wernicke's and Broca's areas.
Exactly. Wernicke's area, usually in the left temporal lobe, is critical for language comprehension. Damage causes comprehension. Aphasia patients might speak fluently, but the words are jumbled, nonsensical word salad, and they can't understand spoken or written language well. They often seem unaware of their deficit.
And Broca's area.
Usually in the left frontal lobe, near the motor cortex. It's crucial for speech production, articulating words. Damage causes expressive aphasia. Patients understand language reasonably well, know what they want to say, but struggle immensely to form the words. It's very frustrating for them.
And damage to the right hemisphere's language areas.
That can lead to aprosodia, difficulty understanding or expressing emotional tone. They might speak in a monotone or fail to pick up on whether someone is joking or serious based on their voice.
Do these differences show up in studies?
Definitely. Studies of split brain patients where the corpus callosum connecting the hemispheres was cut, usually for severe epilepsy, were really revealing.
How so?
If you presented an object to their left hand, which sends info to the right hemisphere, behind a screen, they couldn't name the object because the language centers are in the left hemisphere disconnected. But they could often demonstrate its use or pick it out by touch. It clearly showed the left hemisphere's unique language role.
Fascinating. Okay, let's quickly touch on that clinical spotlight. Head injuries.
Right. A concussion is common, often involves a brief loss of consciousness or amnesia. It's thought to be a temporary disruption of the RES due to rotational forces on the brain. Importantly, standard scans might look normal.
But there are still concerns.
Growing concerns, yes, about persistent problems with memory and concentration, and the potential for cumulative microscopic damage, especially from repeated concussions, potentially increasing risk for later neurodegenerative diseases.
And more severe
Oh,
injuries.
like intracranial hemorrhage bleeding inside the skull. This is dangerous because the skull is rigid. Bleeding compresses brain tissue, potentially causing severe permanent damage. That's why close observation after any head injury is so vital to catch delayed bleeding, and why avoiding a second impact before fully recovering from a concussion is critical risk of dangerous brain swelling.
Right. Important points. Okay, that brings us to the end of our deep dive into this chapter. What a tour.
It really covers a lot of ground, doesn't it? From the basic electrical rhythms of consciousness,
Through the focus of attention, the drives of motivation, the complexities of emotion,
the incredible processes of learning and memory, storing our experiences.
and finally the specialized brain systems that give us language. It's quite the landscape.
It really should give you a new appreciation for the sheer complexity and coordination happening inside your head every single second. Every thought, feeling, action. It's all the result of this amazing biological machine.
It makes you think, doesn't it? As we learn more about how these functions are intertwined, what might that mean for understanding ourselves better, maybe treating brain disorders more effectively? Where do you think the next big insights into the self might come from? Something to ponder.
Indeed. Well, on behalf of the Deep Dive team, thank you for joining us and diving deep into the brain today.